Okay, good evening, everyone. It's a great pleasure to be invited to speak today, and thanks for staying to the end, and I appreciate all the work of the organizers. It's been a terrific meeting. So basically, I'm going to talk about one of the largest brain imaging studies in the world. It's called the ENIGMA Project. And this was an effort to analyze the genetic data of 21,000 people and try and understand some of the main factors that help and harm the brain. So what is ENIGMA? It's a worldwide consortium. We pull brain images and genomic data from a vast range of populations worldwide. The overall goal is to look at, can we find new genes that promote or protect us against disease, genes that affect the brain's wiring, the way that the brain is organized. And this has been a truly global effort. It's one that certainly exemplifies the spirit in OneMind of having so many institutions, so many authors, and such a large-scale sort of crowdsourcing collaboration. And this is run by many, many labs worldwide. The main centers are here at UCLA, also the genetics labs in Queensland, and also in the Netherlands. So why would you need this number of images to find things about genetic risk? Well, the challenge was to find whether single-letter changes in your genome would affect the appearance of your brain on a scan. And we know that these effects are small and that you need a vast statistical database to be able to discover them. You can also do epidemiology with images. So how does exercise, diet, or medication affect the brain? Can you discover some genes that affect the brain's wiring or cause disease? And also, can we estimate our personal risk for mental decline? Now, let me just backtrack a little bit about imaging. Many of the diseases that we know most about, such as Alzheimer's, there isn't a direct way to image the pathology as it spreads in the brain. So we know that Alzheimer's results from beta amyloid and tau protein building up in the brain. And one of the things that ADNI did, I know Stephen mentioned ADNI earlier, was basically give us the tools to look at the physical imprint of these diseases as they spread in the living brain. So this is a movie that we made from ADNI data some time ago that captures 1.5 years of Alzheimer's disease. And you can see the disease spreading. This is in living patients, from the memory centers of the temporal lobe into the executive centers of the frontal lobe, and then well into the emotional centers of the limbic system here. So as you can see, the precision that imaging affords is much more exquisite in its detail than the behavioral assessments that we can do in the clinic from day to day. So this is an excellent target for genetic analysis and also for any kind of query as to what affects the progression of disease. Now, imaging has been accused occasionally of just sort of describing the passive evolution of processes and not interacting. But suppose you had a family member who had schizophrenia. So some of us here have family members with schizophrenia. And they were faced with a choice of taking olanzapine, a second-generation antipsychotic, or an older drug, Haldol, that's been more tried and tested. This was a drug trial with Lilly. It required a vast aggregation of data from many, many patients. And it showed that haloperidol, people taking haloperidol, still lost brain tissue. There was a progressive loss of brain tissue in the frontal lobe, one of the areas that's implicated in schizophrenia. Those who took olanzapine had a lesser loss of brain tissue. Now, this wasn't a report from a single scan. It was an aggregation of a large data set. So can we do epidemiology with images? One of the main factors that we found affects brain integrity and the rate of brain loss as we age is obesity. So there's a deep connection between cardiovascular risk and brain risk. One of the things that we found is that obese people have about 8% more brain atrophy as they age. And this is really a critically important connection between two fields. Now, what about genetics? A couple of years ago, scientists found a gene. Half of us in this room have the bad variant of this gene. And it causes us to crave about 200 more calories a day, causes us to put on about 3 pounds of weight. And you could think that this doesn't matter. I mean, maybe this is something that doesn't contribute to a serious problem. We said earlier that obesity causes a change in the brain. The blood flow to the brain is reduced, the neurons die. We found that there is a physical connection between carrying this FTO gene, again, half of those in this room have this gene, and a loss of brain tissue that puts it at risk for these depredations of brain disorders later in life. Now, how about something more useful? You'll hear in the news very often, Alzheimer's risk gene discovered. And one of the ways that imaging also helps there, and these projects such as ADNI are very useful, is that as soon as a gene is discovered, you can scour databases worldwide for what that gene is doing to the brain. And here's a surprise. So clusterin is a risk gene that ups your risk by about 20% of getting Alzheimer's disease throughout your life. 50 years before the onset of the disease, carriers of this gene, and again, large numbers of us have this gene, show physical changes in the integrity of the brain's wiring. This is an area where David Van Essen's Connectome project will give us huge insight. You wouldn't think that an Alzheimer's gene is already acting 50 years before the disease is being caused. And this seems to be typical. Another schizophrenia risk gene variant, again, this is a large data set that's needed to find the effects of this gene. Carriers of the schizophrenia risk gene, it's called TRAC-A. A tenth of us in this room carry this gene, and they have physically lower wiring integrity in the fibers of the brain, another area that the connectome will give us a great deal of insight into. So based on these findings, we developed a polygenic test to predict your brain integrity. You can have your genome looked at, you can apply to 23andMe or other companies to take a cheek swab and look at your genotype at different locations on the genome. And a lot of the variation in these brain traits is explainable by some of these single nucleotide polymorphisms. So this led to the notion that brain measures may be a very good target for genetic analysis. Now the psychiatrist said, well, you really need about 20,000 people. How are you going to get 20,000 images? The phenotype's expensive. Well, the thing is, there are 20,000 images. You just need to give people the incentive to share them, to aggregate them. There's no need to spend any money. They're already out there. So the notion was screening images for gene effects may be a little easier because it's a much more direct result of the DNA variations that we see. So we formed ENIGMA. ENIGMA is a project, and here's a little animation of where the ENIGMA sites are. It's a consortium of consortia. So ADNI, you see there on the left, already has a huge number of scans. And a great deal of credit should go to Neil Buchholz and those at the NIA, really for making ADNI the model for data sharing in the field. All of these other sites in ENIGMA said, well, we don't have enough data on our own to discover genes that affect the brain. But maybe if we club together and pull this, we can reach that 20,000 mark that's usually needed to see a single gene effect on the brain. So what do we mean by a single letter change in our genetic code affecting the brain? Well, all of us can have our genome sequenced. And there's about 500,000 SNPs or common variants in the genome that at least 1% of us will have. The trouble is, we don't know what a lot of them do. A lot of them may give us a tremendous difference in brain wiring or difference in brain development or a difference in liability to disease. So suppose you measure parts of the brain's wiring, the connectome, or the deposition of amyloid or other types of measure. You can look for associations at every point on the genome. And some of the first genome-wide screens of brain images, everyone said, well, ADNI doesn't have enough images. It only has 800. Is that going to be enough to see any effects? Well, one of the most interesting things is there was a genome-wide hit, a variation in the ADNI subjects that was connected with the volume of a temporal lobe. People that carry this gene had smaller temporal lobes. And it was a variant in the glutamate receptor. And the glutamate receptor was already a drug target for Alzheimer's disease. And so we're beginning to see a connection here between drug targets and genes that affect the physical structure of the brain. Now this gathered momentum. As soon as the next cohort joined ENIGMA, we found a gene that affected the size of the caudate, this major motor nucleus in the brain. This same gene had been implicated in autosomal dominant striatal degeneration, but we all carry a good or a bad version of it and affects the size of this structure. Now, obviously, this needs a lot of work and a lot of scientists collaborating. So 200 scientists from 12 countries could join ENIGMA basically by having DNA and MRI scans from their patients. And we have a sort of scoreboard. You can upload your brain measures and your genomic measures, and we try and aggregate evidence for each of 500,000 genetic variants across the genome to see what they affect in the brain. Do they affect the volumes of parts of the brain, the wiring, the integrity? And 4 Nature Genetics papers came out a month ago. These were the largest brain imaging studies in the world. And there's something very interesting about the data sharing. We coordinated these studies at UCLA, but no genomes came to us. No images came to us. Software went to them. So all those sites that you saw earlier that are in ENIGMA, they said, well, we can't share genomes. Imagine the regulations. We can't give you them. So we sent software out and said, let's have a vote for every location on the genome. Give us the evidence in Germany, Australia, or in Russia that each of these genomic variations affects the volume of part of the brain. This was the volume of the hippocampus. And we found variants that affect brain volume by carrying these variants by a couple of teaspoons full of brain tissue. They affect your IQ by about 1.3 points or so. And a hippocampal volume variant that affects the hippocampus in a very visible way, equivalent to about 3 years of aging. So you could say, well, what about the connectome? David and others this morning gave terrific talks about the connectome. There is evidence to believe that this will be an excellent substrate to find factors that affect brain wiring, if only we pull our data. Now, one of the things that you can see in the detail of these images, they're much better phenotypes than brain scans usually are. I mean, they'll be terrific. let's say you just count the connections. I mean, no serious neuroscientist would stop there, but let's say you just count the connections. This is a map from one single individual with hundreds of thousands of wires or axons that are in it. And let's say we just count the number of connections that interconnect the different parts of the cortex. And David this morning have talked a great deal about the plan to do this on an international scale. One of the things that we did with DTI and ENIGMA is we asked the question, are there any genes that alter the number or proportion of fibers between any regions of the brain? Now, the correction for false positives is astronomical. You're searching 1 million SNPs in the genome. You're searching thousands of fibers. But one of those genes beat the 1 trillion to 1 barrier or hurdle as a gene that might affect the brain's wiring. And this was from screening human connectomes. And it turned out that we could look in ADNI. ADNI was a project that wasn't planned with any intent of being related to the Connectome project. Looking in ADNI, this gene was found to be an Alzheimer's risk gene. It's called SPON1. It's related to amyloid precursor processing. So this sort of serendipitous, but maybe one should say planned discovery, came from aggregating data sets that initially had no intention of being put together. So just to finish off, how can we use this? How can we use these tools now? Well, Dan Geschwin and others at UCLA have a network that aggregates data from autistic children. And one of the enigmas about autism is that there's been no reliable physical record on a brain scan that is consistently found in all people with autism. And maybe the reason is that we haven't been looking with the right techniques. And so this example here is a clinical application of connectomics that's very, very pressing. So Dan Geschwin and his colleagues have been finding genes that increase your risk for autism. They don't cause you to get autism, but in all the maelstrom of factors that add together, they tip the scales in favor of getting it. We found that carriers of these genes have regions of their connectome, the connectivity that we saw earlier, that are more isolated than people without these genes. Those little round circles there, these are connections that are more cut off from the other connections. And so the notion here is that if we screen the connectome, you basically will find hidden patterns in there that even can be linked with single genes, but have a great deal of evidence to understanding the functional organization that leads to these devastating brain disorders. So my acknowledgement is rather long. I will not read the names of the contributors to ENIGMA. But I think one of the notions in these efforts is ENIGMA didn't work in a standard way. Nobody sent a lot of data to UCLA. Software was sent to many countries around the world, and the Germans, the Russians, the Australians, and the North Americans sent back votes for each of the particular genes that was being assessed. And one of the reasons that was effective was that it doesn't cost very much money. Those scans are available. We don't have to collect, if you were asked to review a grant to collect 20,000 scans, no serious person would do it. The scans are already there. And so I think one of the take-home messages is, basically with all the talent and resources that we have available around the world, some synergies could come from integrating this data that we have now, rather than repeatedly requesting more and more data. Thank you.
So we'll go ahead and open it up for questions from the audience. All right, while you guys come to questions, I did want to ask the panel the following, which again, I'm going to draw from Weinberg, who I started with. And he said, we must make big science flourish without, at the same time, allowing it to trample little science. That is, we must nurture small-scale excellence as carefully as we lavish gifts on large-scale spectaculars. And as you panelists have different approaches, generator of large science as well as analyzing it, and aggregators of science shared from many others. Any thoughts on this idea? How do we strike the right balance between these two?
Well, I think one type of science promotes the other. So ENIGMA would never have succeeded without the smaller projects within it. ADNI is a project within it, and that would not have succeeded without the 52 centers around the United States that contributed data. And each of those had talents and expertise that they'd been developing for a long time. Now, one of the ways I think this can help us with making a case for doing these large studies is that we wrote a lot of genetics grants that were small, and they were shot down. They said, you can't do genetics. Any one center could only collect in their lifetime and with their resources about 100 scans. So you can't even start. And I think a means to aggregate data helps these smaller studies get off the ground. You can say, well, I have a population maybe with Tourette. I think Brad Schlagar is here who looks at Tourette. These smaller studies are very, very important, but you have to have the resources to begin them. So this notion of having a consortium is a way, I think, Brad, you'd probably agree, to make these small studies grow, but at least have some incubation period for them to grow.
I guess we've always taken the approach that that's our user base. Those are our customers. So I don't, the individual scientists who are using our data are who we make it for. So I guess to echo Paul's comments, we don't really exist without each other. And I think there's room for both. I think sometimes the conversation is about the zero-sum game, and that giving resources to big science takes away from smaller science, and I think there are good arguments to have very productive programs in each of those.
I think there's evolutionary science and revolutionary science, and the revolutions come both from big science and little science. We don't want to lose the little science, big shifts, that big ideas that come in little groups that won't necessarily come out of big science. On the other hand, some of the big revolutions come out of big science because that's when you reach critical mass to do that. So I think you do need both sides.
Great. Do we have others from the audience? Yes?
So first, thank you. Second, would all of you or any of you just venture how big can big science get? If we end up having mobile biosensors, and when you think of what is already available out there, could we start looking at literally hundreds of thousands, even millions, and put some of these together?
I hope so.
Yeah.
We have some faculty members who are quantified health people, and we're figuring out what to do. You know, how do we put that all together? So, but it's not, you know, it's not that easy. You know, like I know people have spent 1 or 2 decades putting together this huge clinical database, different clinical databases. And at the end of it, they don't discover. Like the saddest day, metaphorically, is when they finish, because then the dream of the great discoveries they're going to make goes away. Because then they hit the reality of it's actually hard to discover things even when you have the database. So even when you have all that quantified self data, it's still going to be hard to discover things.
Yeah, I think that's a great point. There is an initiative that speaks to your comment that NSF and NIH are putting together. It's called the Big Data Initiative. And anyone with so much data that they couldn't ever analyze it is encouraged to apply. In fact, I have to write a grant for this in the next 2 weeks, so it's pressing to me at present. And one of the comments that they made is that in the last 3 years, more digital data has been generated than in the preceding 40,000 years. But the point is, not all of it is useful. So I think a lot of investment will be made on the mathematics and the engineering side to recover information from these techniques, just so we can. One concrete example is, we heard this morning from Olaf Sporns and from David Van Essen. Clearly, the amount of data in the Human Connectome is so rich, you could spend a lifetime analyzing it. But I think a lot of the innovation will come from mathematics and engineering in trying to distill data quickly so that we're not swamped.
So the imaging data kind of brings up a question, and George and I think Brian earlier from the kind of computer bioinformatics side really brought up this issue of data quality. Really turns out to be a major issue, how to evaluate that. And we always hear, one thing about genomics and genetics and things that are kind of commoditized is that it's easier data to share. It's all generated on the very similar platform and all of that. There are a lot of imaging studies that, for example, go on in 20 or 30 or 40 patients in, let's say, schizophrenia or autism, and somebody else does it and finds the opposite result, et cetera.
Sure.
And so I'm wondering how, Paul and others, if you want to comment on this, how one could go ahead kind of prospectively being able to make sense of, of all of that noise.
Sure.
It's there.
Sure. I think that's a great point. We always assume that images are telling us the truth. And nearly every imaging grant has a run-in period where the measures that are proposed are tested for reproducibility, how many patient scans are unusable. And this became a big issue with ADNI. So ADNI intended to scan 800 people, but a lot of them, well, I wouldn't say a lot, maybe 7% of them had stripes in them and artifacts. Many of these were not found for years. And so this is a very interesting example of crowdsourcing. So many people were looking at that data that eventually all of the errors and mistakes in the scans were found and curated. The same with the genomic data for ADNI. Now, one lesson we learned from ADNI was so many people were looking at the data, and I think, Stephen, your point made this as well, when a lot of people are looking at the data set, all of the errors and mistakes get found very quickly. And that's an interesting lesson. There was also an article by Carl Zimmer on the rate of retractions in science is increasing. Well, maybe that's good. Maybe everyone's looking at data much more closely. Maybe a lot of people are finding errors. So I think your point's well taken. I think images do a good job of quality control, but sharing data publicly is a really good way to do quality control because you have a lot of pairs of eyes looking at it, and it makes a lot of sense.